Thank you. I'd like to start by thanking the organizers for inviting me here. This is not a kind of meeting I normally attend, but I'm enjoying it very much to getting really different perspectives on the topics we're discussing. I'd like to also specifically thank Jonas for the superb organization, and I'm pretty sure something's gone wrong in a translation from German to English when it says that he's down as an assistant. I think he's much more than that. So I'd like to unthank Jonas for the impossible task he's giving us to talk. We're not supposed to talk about our own research work, but about a lot of stuff that I really don't know anything about. So, and most of the speakers, of course, ignore this advice, but I'll do a bit of both. So what I thought I'd do is give you a little brief history or overview of quantitative genetics and a quantitative genetics view on heritability or variation in the population, on individual differences, a little bit about quantitative genetics view of gene-environment interactions, and then end with some open questions and caveats and points for discussion. So really we go back to the late 1800s when scientists like famous Francis Galton, who was a cousin of Charles Darwin, started to try to understand the resemblance between relatives for what we call now quantitative traits or complex traits. And this was when the new methodology like correlation and regression was being developed, and Galton applied this to data on quantitative traits in relatives. So here's a famous graph from this paper from 1886, where he's plotted on the y-axis the height of children, and on the x-axis the average of their parents. And what he noticed was the points were not on the line y equals x, but there was what he called a regression towards mediocrity, or a regression towards the mean, we call it now. Yeah, because children here on this side of parents who are taller than the mean in the population are smaller than their children are smaller than their parents, and here the children are taller than the parents who are on average below the mean in the population. We now know that under certain assumptions, and these are assumptions, that the slope of this line is actually an estimate of the heritability of the trait, which is the proportion of total phenotypic variance that's due to genetic factors. And as we can see here, at that time, so that is 130 years ago or so, that slope was 2/3, and as we'll see in a moment, it hasn't changed very much actually. And for those of you interested in not in height, but in traits like intelligence, well, Galton was very much interested in that as well, and was actually quite worried about a regression towards the mean for intelligence. So after this, in 1901, Mendels' laws were rediscovered. And people wanted to apply these kind of laws to think, what does that mean for the traits that we're interested in, like height, intelligence, what have you? So this is a paper from 1903 now, and you can see from the title, this is from Pearson and Lee. You can see it's about the inheritance in man, trying to understand the laws of inheritance. So here's just a simple, you can see now it's a normal distribution, where you take a lot of data on people for height and just plot it. And here's another one of these regressions. And what I've summarized here from that paper is the correlation between first-degree relatives, son, father, daughter, father, etc., and also between spouses. And again, so these correlations are about 0.5 or so, with quite small standard errors. So these were large sample sizes, and the spouse correlation about nearly 0.3. And again, these kind of numbers haven't changed that much since then. If we take a contemporary sample, we get a kind of relative similar values. So what happened at that time, so this is the early 1900s, was a huge debate between a lot of scientists, a lot of big egos were involved. And the question was whether quantitative traits, like height, intelligence, have the same hereditary and evolutionary properties as discrete characters. So it was kind of a debate between, on the one hand, the Mendelians, so that's Mendel's piece, and on the other hand, the biometricians, so people looked at trait like height. So I call this the great Hyde versus P debate of the early 1900s. And it wasn't really resolved until 1918, when a famous paper, or what's now a famous paper from Visscher was published, which basically reconciled the two fields. And this is, to me, the unofficial start of the mature field of quantitative genetics with this paper. So Visscher basically postulated that if you have lots of genes which each individually behave like Mendels' laws, and on top of that you have some environmental variation, that it could explain the observed resemblance between relatives and be consistent with the laws of Mendels' laws of inheritance. There's an awful lot of new stuff in this paper, obviously not easy for us to read because the terminology was all different, but it's, for example, a paper that first introduced the concept of variance. Because Visscher realized that you can add up variances, whereas it's not a very good idea to add up standard deviations. So variance was introduced, and essentially analysis of variance was introduced in this paper. So that's a very brief overview of where things started. And a central concept in quantitative genetics is the parameter heritability. And I mentioned that before. So what is heritability? Well, it's a population parameter. It's the proportion of variation, observed phenotypic variation that's due to genetic factors or due to genetic differences between individuals. It's a parameter that's specific to defined population because specific populations have specific genetic variants that are segregating in that population and also have a specific environment. So it's defined with respect to the genes that are in that population and the environment. It's not just a construct or a concept, it's actually a parameter you can estimate with empirical data, and it's a parameter that can make subsequent predictions that you can test in an experiment. What's heritability not? Because I think often there's a lot of confusion about that. It's not deterministic. I mean, height is an interesting example. The heritability of height is actually quite high, but as we discussed yesterday, height has increased enormously over time. Yeah? There's nothing strange about that, but clearly height, even a high heritability, doesn't mean it's fixed over time. It's not the proportion of a phenotype that's genetic. It's not the proportion of a phenotype that's passed on to the next generation. It's not a parameter that necessarily, or sometimes it may, not necessarily explains between group differences. It's also, and this is a similar argument, not necessarily a parameter that once you estimate it in one parameter, you can also one population, you can also apply it to another population. And it's also not necessarily a parameter that remains the same if the environment changes. So let's look at some empirical data then in how traditionally and in more modern days how we estimate heritability from data. Traditionally that was done, and that's how it was established in the first place, by looking at the resemblance between relatives. So here what I have is on the x-axis here different kinds of relatives which are all first-degree relatives. They all share 50% of their genes by descent. And on the y-axis here, I have the phenotypic correlation again for height. By the way, I use height all the time as an example of a complex trait because, first of all, the heritability is quite high. It's very easy to measure, and you can get extremely large sample sizes, which makes it all very nice model for studying complex traits. So here we have these first-degree relatives, and you can see the phenotypic correlation is quite similar between all of these. And these are for 4 different data sets which span more than a century worth of time. So this includes Pearson and Lee from 1903, and these other 3 are more contemporary data. Usually when we try to estimate heritability, we don't want to rely just on one kind of relative, like just having full SIPs, for example, brothers and sisters, because obviously there can be some confounding with environmental covariance, or as it was mentioned yesterday, shared environmental factors. So usually what we do is we have different kinds of relatives, and I've shown that over here. So on the x-axis now I have genetic relatedness, whereas 1 means monozygotic twins, genetic relatedness of 1. a half are these guys over here, a quarter are, for example, half SIPs or individuals and their grandparents, and so on. And then we have a quarter and zero, sort of unrelated. And these, when you put the data points there, and each of these points is based on very many different pairs from these data sets here. What you see it's a very nice straight line that you can easily draw through the points, and with a slope that again is consistent, but doesn't prove, but is consistent with the majority of phenotypic variation for height being genetic. It's about 3/4 here. And by the way, the linearity here also suggests that most of this genetic variation is additive by nature, because otherwise we'd see strong curvature there. We can also just look at a twin study. This is data from my colleagues in the Queensland Institute of Medical Research, Nick Martin and colleagues. And very large data sets. Here are just a correlation of male monozygotic twins, just plotted one against the other, correlation 0.85. And here are male non-identical twins, dizygotic twins, 850 pairs, correlation 0.45. And under some assumptions, and again I stress that these are assumptions, you just take twice the difference and you get an estimate of heritability, and it happens to come out at 80% from this data set. So all the observations on the resemblance between relatives are consistent with a high heritability for height. So what about other traits? Because height is just an example. Well, last year, a number of colleagues and I reviewed all the twin studies ever done. And this was a lot of work. I hasten to say most of the work was actually done by Daniel Postuma. And what this paper just showed was when you just look at all these correlations that are reported in the literature from a lot of different traits. These include a lot of behavioral genetic traits. They include also a lot of diseases. That majority of the traits, but clearly not all, are actually consistent with an extremely simple hypothesis. The majority of the traits are consistent with the hypothesis that all the resemblance between twins are genetic and that most of genetic variation is additive. That's the conclusion. It doesn't, of course, prove it. You could have lots of more complicated models where things cancel each other out, but that's the most parsimonious explanation of the data. But it's clearly not the case for every individual trait. There are plenty of examples where this simple model doesn't fit, but that's what we found. Now, that's from the relatives. What we're in now is a genomics era where we can actually start to dissect this genetic variation that we believe is there in the population, because we've estimated it from pedigree or twin data, to dissect that variation in contributions from individual loci. Now, again, height as an example, there are clearly Mendelian forms or pseudo-Mendelian forms of height, for example, forms of dwarfism, which are caused by gene defects. There are forms of particular Marfan syndrome, where individuals are taller than average in the population, again caused by a rare mutation. But by and large, if we look at the distribution of height in the population, and we look at the resemblance between relatives, etc., the conclusion is, and that's without actually finding genes, the conclusion is there must be lots of genes with small effects. You know, because we have nice normal distributions here of height, and the heritability we know is high. That's not consistent with the lots of genes like that being segregating in the population. So it was mentioned a few times yesterday, these Genomewide association studies that have been around for about 10 years now, and they have been an absolute game changer in the study of complex trait, not just behavioral traits, but also in particular disease. So the era before that, which was characterized by candidate gene studies and linkage studies, basically were not a success. I think it's fair to say that. But when these studies came along, which is all driven by technology, a lot of discoveries have been made. And this is just showing some examples of, this is not over time, this is just to do with sample size, But you can also think about it as a timeline here, where this would be about 10 years ago or so, where for a lot of these diseases that people knew from studying risk to relatives that there must be genetic variation there, but it was very difficult to find individual genes. That through genome-wide association studies, there are now hundreds for some traits, hundreds of robustly associated DNA variants that people can follow up for function and maybe ultimately think about new treatments. And height's there as well. So again, I keep coming back to height because it's such a well-studied trait. It's a byproduct of lots of medical studies. You can get huge sample sizes. So I'm going to summarize what we know about height now. So here we have these bars represent variation. So we've got 100% variation, which is total variation we can observe in a population, so due to both genetic and environmental factors. We have about 80% that we believe from the pedigree and twin studies, etc., that are due to all genetic variants combined, so that's the total heritability. We have 16% now of variants that are captured from individual loci. Those are about 700 loci that are now robustly associated with height. And we have 45% of the variation that is actually captured by one of these SNP arrays that are used in genome-wide association studies. If you were to go, and that's basically common variation everywhere in the genome, if you were to go from a SNP array to more like full sequence data, that's something in between, which is sort of imputed sequence data, but if you go through that, you recover nearly all the genetic variation. So in terms of the missing heritability for height at least, there's a very small gap now between what's been captured in totality from a think about a genome sequence or an imputed genome sequence and the total heritability, which is all genetic variation that's segregating in the population. Now, of course, I know you're not interested in just, or not interested at all maybe in height. Try to summarize here when we bring in other traits, for example, IQ and schooling as a trait, number of years of school you went to, educational attainment. The heritability estimated from pedigree data or from twin data for height, as we saw, is about 70–80%. I'd say for IQ about 50–70%, although Matt Magoo made a point yesterday that might be actually higher if you look at twin studies. And for educational attainment, I reckon it's about 30–40% from some of those studies. The heritability that's captured from the whole genome, so that's captured from associations between common DNA variants and the unknown variants that are causing the effect, it's about 3/4 for height, about 50% for IQ, and about 50% for educational attainment. This is just an estimate of how much is captured from the genome. That is not to say that we found all the genes, because that's a very different picture. If we look at the sample sizes from these genome-wide association studies, you can see that the traits that are easy to measure, which is how tall are you or how many years did you go to school, have a much larger sample size than something like that needs a psychometric test like IQ. The genes found to date for height with this sample size, as I said, is 700. I think it's only one for IQ, which is the ApoE, or a variant of the ApoE Tom40 locus, which I think is usually associated. I'm sure we can debate about whether it should be 0, 1, or 2. And about 70 for educational attainment. That's a paper led by Philip Kullinger, who's here at the back, that's coming out hopefully soon, or will be published soon. And if we look at how much of the heritability is captured from these known loci, it's about 20% for height. That's basically now, you could say, is due to variants that are robustly specific genes that we know about, and we can go to a new sample from the population and replicate that result very robustly. Less than 1% for IQ, and I think about 1 or 2% for educational attainment. Now the workhorse, we'll come to modeling, and I might as well show an equation. I think this is the only one I have. Sort of the workhorse of quantitative genetics is an extremely simple equation. It's basically saying that the phenotype that we can observe is due to genetic factors and environmental factors. And at this point in time, I'll just sum them up. Now, what are these things? And I think there's been a lot of confusion about that in the literature, and some discussions yesterday also point to that. So G is basically the effect of the whole genome on a trait. In the past, we inferred G from just looking at pedigrees of families, and in the last sort of 5-10 years, we can also do that for SNP data. G is in principle very easy to measure. You just need a DNA sample. With the widespread availability of data on these SNP chips, there is an increasing amount of research we can do centered around G. This is not to be confused with little g for used in intelligence studies. E is the effect environmental factors, but it's the effect both of, in this case, the way I phrase it, of both unmeasured and measured environmental factors. And usually, because there's not an awful lot of environmental factors that we actually measure, usually in statistical analysis that just is inferred just from variance partitioning, how much noise is there. But if we have measured effects, of course, we can just do estimate the effect size. Now, one thing that was mentioned yesterday, I think in the first talk yesterday, was a GE covariance or correlation. And that basically means that in the population, they're correlated. And I think, and this is a point for discussion later, that it's probably very common for many common traits, in particular the traits that have been studied here. So there's a confounding between G and E. And clearly parents pass on, if we think about transgenerational effects, parents pass on genes, and those genes have effects on traits, but they pass on also something about their environment. So if these things are correlated, they're also passed on in a correlated fashion to the next generation. If we think about most of the statistical modeling that we do with empirical data, in most cases, unless we model this covariance explicitly, it actually ends up in G. If it's confounded, it ends up and it looks like genetic variation. And if the confounding between the genes and the environment is not changing because the environment, if the environment were to remain the same, then it is basically genetic variation. Because think about heritability, it's defined with respect to environment. And if in that environment the genes and the environment are correlated, then it looks like G. An interaction between genes and the environment basically means there's a difference in the magnitude of direction of the effect of an environmental effect or exposure on the trait in people with different genotypes, or you can also phrase that the other way around. And because there's an interaction, it immediately means it depends on the scale. If something is additive on one scale, then it will be so no interaction on one scale, and you transform that scale, for example, from additive to multiplicative, then you will find, per definition, you will find an interaction. So that makes it extremely. You have to be extremely careful when you talk about there's an interaction, whether that's scale dependent, what the scale of interest really is, and whether if a simple transformation makes it go away. You have to think hard whether you still call that an interaction or not. And unlike the GE covariance or correlation, if you don't model G by E interactions explicitly, it usually ends up in the environmental part. So if there were to be widespread evidence for genotype and environment interactions, then it would predict that the heritability of the trait would be would basically be lower than in the absence of genotype and environment interactions. Because it goes into the residual or in the environmental part when you partition phenotypic variation. G by E interactions, as we've heard at this workshop, is very much researched, and I would say it's also very much hyped. Yet, to my knowledge, if we look at most hard empirical data, it suggests that it's not important in explaining variation in the population. I'm not saying it's not important and that these effects don't exist. I am interested in how much variation does it create or variation does it explain in the population. So a G by E effect is different from how much variance it explains. It's exactly the same argument as genotype by genotype interactions. Yeah, so epistasis. It's exactly the same argument, that clearly there are molecular interactions between molecules, and you can find them if you have the right data, but it doesn't mean that it explains individual differences in the population. So I now come to just a bunch of comments and queries and points for the discussion. So my guess is that the environmental part is just as multifactorial as G. We know that from the genetic studies over the last 10 years, most traits we look at, including behavioral phenotypes, there's probably hundreds, if not thousands, of genetic variants underlying those that explain or that underlie the observed resemblance between relatives. My guess is that the environmental factors are just the same, and the question is, how do we measure that? You know, what is the right measurement if we were interested in dissecting environmental variation? Because if we dissect both genetic and environmental variation, we can, of course, explain the total phenotype better. I don't know how to do that, but you know, one idea is could you know, smart devices, you know, where you measure people continuously on a whole lot of environments they're exposing all the time, whether that's one way to think about, you know, getting dissecting environmental effects. I've no idea, something for the discussion. Another question, which I think is more tangible and in the short term, is how can we use or leverage the information that's now available from genomic data to answer questions in the social sciences? And I think one of them is very mundane. It's basically saying, if you're in your experiment are interested in, say, treatment effects or differences between two groups, and you were to have a DNA sample on those individuals, you can account for some of the individual differences already. So you actually have a more powerful experiment if you adjust or account for the individual differences that you know from outside information explains some of the variation in your experiment. That's kind of you know, mundane. For example, for height, if someone was interested in the effect of some environmental exposure on stunted growth on height, if you could now, in principle, explain about, you know, between 15 and 20% of individual differences from the genetic factors that have been identified. So you could basically have the power of your experiment basically would be increased in finding treatment effects or differences in the exposures. I think it's very exciting that this whole field of genomics brings with it new experimental designs, because basically what is happening, if all of us here had their DNA measured on one of these SNP chips, we suddenly all can be linked together in almost a randomized experiment, because some of us will share more of that DNA in the genome than some pairs will share more than others, and we know that actually explains some of the differences. So we can actually suddenly have almost like a huge randomized experiment where everybody is linked. And the cool thing about that is that's very unlikely to be environmental or shared environmental factors between us, for example. And I'll show you some examples of that, tangible examples, in a moment. Also, what's exciting about this, if you had a DNA sample and you did not have a particular trait or covariate measured, you can actually impute it to a certain extent by using just the genetic predictor of the trait. And again, those it's early days for those, because a lot of those predictors are not very powerful. But I think more, sample sizes are still going up dramatically, and I think that will improve over time. And finally, I think there are also ways to use instrument variable analysis to start thinking about inferring causality. And I know that's a loaded word, but I'll just give you an example from the medical field in a moment. So here is an example of a new experimental design where we asked the question whether the trait height or body mass index or waist-hip ratio, which has to do with the fat deposition in the body, whether these traits are actually the same traits in males and females. And so these clearly, so these are data sets, as you can see, large data sets here, where the individuals are unrelated to each other. They're just a sample from the population used for genome-wide association study, but we can utilize those data to answer these specific questions. And basically what we found that for most of the traits, that's for height and body mass index, it's an estimate of the genetic correlation. If that's 1, it's basically saying there's no G by E, where E here is whether you're a male or female. For most traits, this estimate was no different from 1, suggesting there's just the same trait there in males and females, apart from waist-hip ratio adjusted for BMI. And that's a trait that's also, for different experiments, has been shown to show differences between males and females in terms of where, particularly in obesity, where the fat is deposited in the body. And we found an estimate of 0.6 there, and that was different from 1. So these kind of experiments would not have been possible in the past. In the past, you would have to rely on, for example, a twin study or an extended pedigree, and that comes with additional assumptions about shared environment and so on. So in a way, these experiments are cleaner because they are on different people. Here's another one with two examples here. On the left-hand side is an example from the medical genetics, where exactly the same kind of idea was used to estimate the genetic overlap or comorbidity, if you want to think about it that way, of different psychiatric diseases. So here's a bunch of psychiatric disorders: ADHD, bipolar, schizophrenia, major depression, and autism. And here is an estimate, essentially what's called co-heritabilities, which is just a scaled genetic covariance. It's essentially the genetic correlation times the square root of the 2 heritabilities that you capture from the SNPs. And these are experiments on different people. These are case control studies on one disease, case control studies of another disease, no individuals in common, but the SNP data linked them, and you can exploit that to estimate these parameters. And what this found was a surprising sort of overlap between schizophrenia, for example, and major depression that people had talked about, but there was no hard data to show that. So this was quite an interesting way of exploiting the availability of these data to answer questions about genetic overlap. It also shows that the whole, I'm obviously not a psychiatrist, but this whole pigeonholing of people in terms of this, that, or the other doesn't make a lot of sense, because there's clear overlap between a lot of these traits, at least from a biological point of view. You can also do that with traits that might interest you, more non-medical traits. So here's an example it was just published in Molecular Psychiatry, work from a collaboration between our colleagues in Edinburgh and ourselves, which is different methodologies to try to estimate whether there's a genetic overlap between genes that are associated with cognition, with IQ, and with body mass index. And basically the bottom line is here, these are all from different kinds of experiments. This is 2 different data sets where so this is all GWAS data, where the same people were measured for BMI and for IQ. The second line here is an estimate where the data on body mass index was completely separate from the data on which IQ was measured. So definitely no shared environmental factors there. And the third one is from a new analysis that relies on summary statistics that are in the public domain on these traits. And the bottom line is that although the standard errors for some of them are large, all of them give an estimate of genetic correlation between these two traits, which says that if you have genes that are associated with increased IQ, then the same genes are also associated with a decrease in body mass index. And we can come back to that, what that means. And the final example of a new experimental design is what I mentioned about instrument variable analysis. This is actually quite a nice paper. It was published in The Lancet a number of years ago now, which exploited the fact that there were a number of these genes identified that explain differences in cholesterol. Now, cholesterol is associated with heart disease, myocardial infarction, and the question is whether the observational data is also points to a causal mechanism. So, for example, the observation is, from observational data, that individuals with higher sort of bad cholesterol have an increased risk of infarction, whereas individuals with higher levels of the good cholesterol have a lower risk. And the question is, are there confounders here, right? Do these particular observations imply that there's a causal mechanism there? So basically what these authors did, they used a predicted genetic effect, so a genetically raised sort of LDL cholesterol, that's the bad cholesterol, or a genetically raised, so that's the instrument, good cholesterol, and then looked whether that prediction was also associated with increased risk. And they found, yes, it is for the LDL cholesterol, but no, it is not for the HDL cholesterol. So the policy at the time in a number of countries and a lot of doctors would be, you have to increase your good cholesterol, whereas this kind of analysis suggests, no, the policy from a medical point of view should be to lower your LDL cholesterol, because that's really bad for you, but it doesn't help you to increase this one. So this observation is probably caused by some confounder that you haven't measured. This is just one example, but you can also use that in kind of fields that we're interested in. So some questions I think for later on is whether this sort of social influence is an environmental effect, or should we just treat it as a trait, as a phenotype? For example, socioeconomic status or educational attainment. As I've shown you some results where basically educational attainment was treated as a trait and not as an environment. And this is maybe thinking too far in advance, but for genetic effects, what we can do, because of all this genetic data that links us, we can create a kind of relationship matrix between everybody in terms of how much do we share. And that can be used for a lot of analysis and to try to explain variation. Can we do the same for the environment, and what would it look like? What is the equivalent of some environmental relationship matrix? And can we ever measure it? Because I think that would be really helpful in trying to dissect the total variation that we can observe into, or to quantify that into components of variation or sources. And finally, I want to just finish with my view on the discussion yesterday that took place about candidate genes, etc., and interaction effects. And this is just I'm a simple guy. There's very simple reasoning here. So I think, at least in my field, most people would agree with the statement that candidate genes for complex traits, that includes a lot of disease studies, were basically a failure. They've not led to robust associations that you could find in a new sample, and that would lead, for example, to drug development or whatever. Genome-wide association studies, on the sort of in contrast, have provided robust evidence that there are individual genetic variants associated with a trait. And if you take a new sample from the population, again, if you're sufficiently powered, you find these effects. They're real, and it's already actually a lot of biology that's taking place to nail down, what the likely causative variant is in the genome that is responsible for the observation that you can, the observations. But because there's so many genes, like for height we've got 700, it only explains 16% or about 20% of the genetic variation, right? So there's many, many more. So clearly the individual effect sizes are tiny, they are small. Therefore you need very, very large sample sizes to detect new variants. Genotype by environment interaction studies, or studies of variability, and we've done some, as I mentioned yesterday, ourselves, or sensitivity or plasticity, whatever you want to call it, they are studies of interactions. They are, per definition, studies of second-order effects. And we know if you want to detect an interaction, you require a larger sample size than detection of main effects. Yeah? So then, if there are lots of so-called replicated G by E effects that are reported from studies with smaller sample size than G was, and also used upon candidate genes, it just doesn't fit with the observations that we have on much larger sample sizes. That's all the point I want to make, that there's something which basically is not consistent there. And I'll stop there. Thank you.